Good morning, everyone. My name is Dr. Natarajan Ranganathan. They call me Rangan or Dr. Rangan. And me and Dr. Miller, I think, probably belong to the same generation in trying to think gut microbiome and probiotics outside the box, because normally probiotics are known for digestive health or gut health, but he related to more like obesity, how it can be. Now I'm trying to just take you into a different field, but I'm not going to go very much into the probiotics or definitions or whatever it is, because I did have a 15-minute presentation on Monday. So I'm going to run through some of the slides to take and see how the dysbiosis, what we mean by dysbiosis is the imbalance of the gut microbiota, I should say, causes chronic kidney disease. Not only chronic kidney disease, how the imbalance of the gut dysbiosis is related to various disease, but how we have taken that to the chronic kidney disease. Well, I just don't want to go through this. I'm going to run through this because we have already gone through. The only thing I want to tell you is, can you see this? This was in 1999 in Scotland. There was a symposium of the International Society of Artificial Internal Organs in Edinburgh, and we were four of us. The gentleman on the right is Dr. Carl Schellstrand from Karolinska Institute in Sweden. And then the next one is Dr. Tatsumo Sonaka from Japan. And of course, you see as me, if you want to be an entrepreneur, you can really lose air. You can see the difference between that and now. And the last one is Dr. Eli Friedman, who is a very distinguished professor of nephrology at Downstate Medical Center. So here I am, a synthetic bioorganic chemist, not knowing anything about probiotics or anything like that. The only concept we had was, we have nephrologists, and I am medical research and organic chemistry. Our concept was, we didn't know anything about the gut. The only thing is, one thing we know, that when the kidney failure happens, there is a diffusion of uremic toxin from the circulating blood into the bowel. So our idea was, can we use a bacteria or sorbents and remove the uremic toxin via the bowel? That is why we just thought, can the bowel substitute, will the bowel be the kidney of the future symposium? And you can see this is in 1998. I told you, these are all reactions happen in your body. And whatever research paper you see is just only little. We don't look into the complete aspect as you just see holistically issues in the ground. I'm going to run through to give you that we just distinctly know that all diseases are related to the dysbiosis of the gut microbiota, and we are actually going into what we call as genomics, transcriptomics, proteomics, metabolomics, epigenomics. All we are going to omics. I did describe what we mean by omics. I don't want to go through this again, and just want to give you an idea. If you just take your whole body and count the number of cells that is the whole body has, the total number of cells that is present in the whole body is about 10 trillion cells, I should say. And if you take just the gut out of it and find out how much the gut has, the 2 kilograms of the gut that you have is over 100 trillion cells, and then the number of genes is 3.3 million. So it is only at 2004, we all recognize gut is an organ. Before that, nobody recognized gut is an organ. They used to think liver is an organ, the brain is an organ, breast is an organ, kidney is an organ, but we never thought gut is an organ. Now we're realizing not only is an organ, it is an extremely important organ, because we have discovered so much about what the gut has, I should say. Now, this is all possible because of the sequencing technology that has taken place, and we have gone through this, and we have gone through its probiotics, how we define it, and we told about probiotics and prebiotics. But what I just wanted to just start with is just to give you some sort of an idea. Yeah, how does the gut microbiome? It has 3 different properties. That is, the gut microbiome is what we call immunomodulatory properties. It controls inflammation of bowel diseases, it strengthens innate immunity, alleviates food allergy, and this is one of the aspects of the gut. The next one is it normalizes the intestinal microbiota. That is why we are talking whether we can normalize it by feeding a good bacteria whenever there is an imbalance. And last but not least, it has a neural effect. It is neural behavior, mood changes, changes in synaptic plasticity, and all these sort of things. And now only we are learning that there is distinctly various probiotics that has been in the market. They're all dietary supplement for irritable bowel syndrome, ulcerative colitis, pouchitis, oral derivatives, vaccines, colic pain, and all other things. But again, we never thought that the gut microbiota is related to kidney disease. It is actually, we went through that. He already told about taking this lean microbiome and put it, the lean mice and put it under germ-free conditions into this one. It becomes lean, and we also saw this, that the microbiome, he already said about that. So what we are just thinking, obesity, of course, we also clearly showed that obesity is related to the imbalance of the gut microbiome. And obesity, why it is related to gut microbiome, is what we started with, because 1/2 of the obesity patients approximately end up with kidney failure. 1/3 of the patients of hypertension will end up with kidney failure. The remaining is from HIV, lupus, and tuberculosis, etc. So our interest centered around, because of the chronic kidney disease, the cause of chronic kidney disease is obesity, hypertension, and infections like tuberculosis, etc. But we never ever had a connection that these diseases are related to the dysbiosis or imbalance of the gut microbiome. So dysbiosis is nothing but imbalance of the microbiome, gut microbiome. Normally, they say if you have about 85% of the good microbacteria, gut microbiome bacteria, bifidogenic as well as lactogenic, it can sustain about 15% of pathogenic bacteria, I should say. So this is sort of balance. If this balance is tilted, I should say, then you come across all sorts of diseases. Now it is becoming very distinctly apparent that all diseases like chronic kidney disease, brain dysfunction, asthma, hypertension, colon cancer, obesity, everything is related to nothing but it's all related to the imbalance of the gut microbiota. However, what we have done is we are addressing to chronic kidney disease. And we do have 2 products. One is called as Ranganathan, which is for human applications. The other one is called as Azadin. So I'm going to take you the story how we developed this product, I should say, how we got centered into this. Now, if you take ordinary kidney, what does a kidney do? The blood passes through the kidney, and the unwanted nitrogenous substances like urea, uric acid, creatinine, it is excreted, and the clean blood is returned back to circulation. That's what your normal kidney does. However, when the kidney is impaired, this doesn't happen. Your urea, uric acid, creatinine, or nitrogenous wakes, these are the toxins. The toxins increase. As the toxin increase, it is called as uremic syndrome, and it becomes fatal if not treated. The old concept was, it was always thought it is only urea, uric acid, and creatinine. But now we are learning there are over 100 uremic toxins, some of them present in nanomolar and picomolar concentration, which are extremely highly toxic actually. But we never took a look at it, because now with the advances, now we can analyze what are all the toxins. Especially if you see the toxins that is retained besides urea, uric acid, creatinine, you see this indoxyl sulfate, phenol, paracresol, beta-dicarbonate. These are all highly toxic, present in a very small concentration, and the lab report does not report the level of these toxins, I should say. So we decided, well, can we in a position try to take this advantage of it? The reason why, if you see, you can see this paper came in 1973 by Spark, who just published. If you see the mean distribution of some of the uremic solutes, that is in the gastric, bile, as well as intestinal region, the intestine has the large amount, the large bowel has the large amount of the toxins. So we are certainly very rational, logical in trying to address the removal of uremic toxin using the bowel. At this point, I will digress and give you some idea about kidney disease. There are 5 stages of kidney disease. Actually, you can live with one kidney. That is why sometimes you see ad, don't go to heaven with a kidney, donate it, just as I said, no, donate the poop, poop is free, we are taking the kidney is going to be more expensive. If you have even 50% of the kidney function, you are perfectly fine. But if you lose 10% more, 50 to 60% is called as stage 1, 60 to 70% kidney function loss is called as stage 2, 70 to 77.5 is called as stage 3, 77.5 to 85 is called as stage 4. They are normally reported as GFR, glomerular filtration rate, but I'm using the word percentage to make you more conversant, as you know. So, up to 85% kidney function loss, for 50 to 85, stage-wise it happens, and this is as the result of a primary disease. The primary disease could be either hypertension, or diabetes, or tuberculosis, HIV, etc. But by the time they find out that you have a kidney problem, you are already in stage 3 and stage 4, because the lab report tells always over 60, I should say. By the time they find out you have kidney disease, you are stage 3 and 4. So these are what we call as a pre-dialysis patients. That is, patients whose kidneys are failing but not yet reached the stage of dialysis. But when once you reach 85% or greater, at that point you have no other option other than going in for a transplant or going in for a dialysis. Dialysis is hemodialysis 3 times a week, 4 hours per session. And in USA, there are about 500,000 patients. The insurance company bears the cost of it only for about up to maximum 6 months to a year. After that, it is taken up by the US government. US government spends close to about $80,000 per patient per year actually on dialysis, and it's almost $35 to $40 billion that is spent by the government in treating the dialysis patients. But equally, there are stage 3 and stage 4. To delay or postpone the need for dialysis, they use various type of medications like ACE inhibitors or phosphate binders, etc. And you spend about $25 billion market. So we actually spend close to about, for kidney disease, in talking USA, I should say, about $60 billion. So our idea was, can we delay or postpone the need for taking this pre-dialysis patient to reach the dialysis state? They may be in 2, 3, or 4 years, but can we delay it as long as possible? The second goal was, can we reduce the duration or frequency of dialysis? Instead of 3 times, can we make it 2 times? Or instead of 4 hours, can we make it 3 hours or 2 hours? So that was the object of doing this. So if you take this one, you can see this only in 2012. We distinctly reported by Dr. Waziri that the dysbiosis is related to chronic kidney disease. Until such time, there was no proof that chronic kidney disease is related to the gut microbiome. But we thought about it in 1998, okay? But we didn't know it is the dysbiosis that is happening, I should say. So you have got good healthy bacteria like high levels of lactobacillus, high levels of bifidobacteria, that is in a healthy population. But when you have this CKD or chronic kidney disease, for that any disease, there is an accumulation of uremic toxins. Now these uremic toxins, as they accumulate, there is a low level of lactobacillus or bifidobacteria. So because there is a larger amount of pathogenic bacteria, these bacteria actually, what they do is not only grow in the large bowel, they start growing towards the small bowel. We call this small bowel bacterial overgrowth. Now, we are finding out, not necessarily in kidney failure, this was reported in 1996, small bowel bacterial overgrowth. Now, we call this SIBO, small intestinal bacterial overgrowth. Not necessarily in kidney disease, other diseases also, they have this small intestinal bacterial overgrowth. And these bacteria, because they are growing, they actually rob the nutrients or diet that you are already taking. So the people don't get much of a nutrition, the patients don't get much of a nutrition. they feel weak, and the quality of life of these patients are very poor. So we thought we should be able to add a good bacteria, I should say, so that we can remove the uremic, the pathogenic bacteria, or at least to a certain extent balance it to a greater level. As I told you, Azadil is our wet product. I just want to give you an idea. This was in 2005. We got about $3.25 million grants, from NIH, USAID, and qualifying therapeutics. When we exhausted all our grants, we were looking for money. Nobody wanted to give us money because our product is a dietary supplement. One of the venture capital lady, she didn't give money. She said, Dr. Rangan, why did you sell your products to cats and dogs? Because cats and dogs also have kidney failure. We never expected that. So what we did was we put our product on sale on the internet. That time we didn't have this Google or Yahoo or anything like that. If you remember, what did we have that time? 2005, 2006. It was a chat room. Our top product became a chat room conversation. When it became a chat room conversation, you may be surprised, I should say. It caught fire like anything. And then there were 3 companies actually asking us to license the product. We actually gave it to one of the French company, what calls Vetacanol, and that is how we were able to survive. So even today I say our company was saved because of cats and dogs, I should say. And I'm not going through, but we have published this completely, whatever we have. Now, our product that we have is what we call as Renadil. It has 3 distinct microbes. It has what we call as Streptococcus thermophilus. Streptococcus thermophilus removes urea, uric acid, creatinine. We have got what we call the Lactobacillus acidophilus. It removes methylamine, dimethylamine, carcinolamine, trimethylamine oxide. That's also one of the things that it removes, as well as we have bifidobacteria, which removes indoles, phenols, and some of the aromatic metabolites. So it is a group of these 3, and we have this product, what we call Renadil, and it is 45 billion per gel cap, and we actually sell it online, I should say. So we have a product for cats and dogs, smaller capsule. We have a product for Renadil, which is for human product, I should know. You may be surprised. We have taken this to almost like a pharma-like validation. How we started was, we started taking an artificial intestinal fluid, and into the artificial intestinal fluid, according to US Pharmacopoeia, what we do is we add uremic toxin, urea, uric acid, and creatinine, and we used to add individually or collectively these microbes. To begin with, the flask will be totally crystal clear, transparent. But as time progresses, we will take an aliquot of it, measure the amount of the uremic toxin that we added. Within 6 to 12 hours, whatever toxins we've added is gone. Where did it go? The flask to begin with is totally transparent. But as time progresses, it becomes slightly turbid, white, milky white, and by measuring the optical density, we demonstrated the growth of these microbes is because of the utilization of the uremic toxins, and we were able to actually culture that and also identify, yes, these are the things that is responsible for what we have added, and we were able to prove that it is the growth of the microbe is because of the utilization of uremic toxins. Then what we did was, what we have done, we used a machine which mimics our gastrointestinal system. It is a 5-step biochemical reactor, one representing stomach, one representing the small intestine, one ascending colon, one transfer colon, one descending colon. It is called a simulated human intestinal microbial ecosystem machine. This machine is headspace control, pH control, temperature control, even the peristaltic movement of the gut control. Using this machine, we demonstrated by putting these 3 microbes, the question came, are they still alive or are they fighting with each other? Second, do they individually and collectively remove the uremic toxin? Third, the question came that do they disturb the microbial ecology that you already have? That time we didn't know much about the gut microbiome, but we wanted to do this stuff. All these 3 answers we affirmed, confirmed by using this by 1 year of collaborative work with Dr. Willy Verstadt at Ghent, Belgium. Yes, when you put these 3 microbes, they are still hibernating. They are still alive. Second, they are actually individually and collectively removing the aromatic toxins and producing the short-chain fatty acids. Third, we also demonstrated they do not disturb the microbial equilibrium that you already have. In addition to that, a question came, how do you know that by utilizing, because on average we produce about 15 to 25 grams of urea per day. 15 to 25 grams of urea per day, if you produce, where is it going? Urea is hydrolyzed to ammonia. Ammonia is very highly hepatotoxic. So what we did was, even in our in vitro experiments, we actually put an ammonia and electrode, as you say. And we demonstrated when we have the microbes, the ammonia produces 50 to 60 parts per million. But at the same time, if you take just a pure urea, add a urease enzyme, and look at the ammonia produced, it's over 70 to 80,000 parts per million. And in this machine, same machine also we demonstrated, there is no ammonia produced, as you say. And the explanation for that is ammonia is immediately transplanted, transaminated in the alpha-ketonic acids to form amino acids. Amino acids form dipeptide, polypeptides, and proteins, and it is the protein of the microbes that you are just seeing in the feces. So we distinctly demonstrated that. After that, what happened was we used to use rats, which is 5-6 nephrectomy rat. You just take a rat, remove one kidney, and remove 2/3 of the other kidney. It is 5-6 nephrectomized. It is about stage 5. They normally survive for about 40 to 50 days. With our product, they have survived over 145 days. Not only they survived 145 days, we could have survived longer, but it was during Thanksgiving time. There was nobody to take care of this. So before going for Thanksgiving, actually we killed all 172 rats by putting them in a CO2 incubator. Although we did studies with the rats, there was a criticism. Rats' gut doesn't mimic ours, I should say. Then we found out pigs. Pigs are much more very similar to our physiology, but the pigs are very huge to handle. But we found out a particular type of pigs called as mini pigs. These mini pigs are called as Göttingen mini pigs. They are from Germany. So what we did was we imported 2 sets of mini pigs, 2 males and 2 females, and actually reared them in a farm in Lancaster. As the piglets came, we transferred them to Indianapolis, Indiana. There the transplant surgeon did 5-6 and by putting our product, we were able to show the level of urea and creatinine in these pigs come down. Not only that, the pigs are so active, you would not be able to know which is a placebo pig, which is a pig that has been given our product. After that, of course, we did some human clinical trials, and I told you we just started selling the product, and more or less we have completed this. And we also did some human clinical trials in Canada, Mexico, Argentina, and also Nigeria, and we have published all this. So we have multi-site CKD 3 and 4. We have dose escalation studies. We have studies in ESRD studies, and we have also done a consumer survey. And all of this is published, and if you just go through our website and look into that, you'll be able to see all that we just published. So not only that, last year we put it in the Harvard Medical Symposium, all the data of all the clinical trials. And then one of the gentlemen from New York Academy of Sciences who attended that went and told the president that this work needs to be exposed. So they called us. Normally, when you present in one abstract, you don't do that again in some other place. So we presented this also again New York Academy of Sciences, and recently we presented this in the month of April in the World Congress of Nephrology, because everybody wanted to know what work we are doing, what scientific we do. So kidney has 3 different functions. One is a secretory function. It secretes erythropoietin for red blood synthesis, vitamin D for calcium metabolism, retinine for control of blood pressure. It has a regulatory function, regulation and maintenance of homeostasis of sodium potassium phosphate. It has an excretory function. You know, all these functions are in a dynamic equilibrium. So any disturbance in one will affect the other 2. At the present time, most of the nephrologists treat only the symptoms. If it's anemic, they give erythropoietin. If there is any problem with vitamin D or calcium metabolism, they give vitamin D. If there is any problem with the blood pressure, instead of renin, they give ACE or ARB inhibitors. If there is too much of phosphate, they give phosphate binders. If it is too much of potassium, they give potassium binders. And to increase the urea output, they give what we call as diuretics. So they address only the symptoms. But as a holistic thing, we continuously remove the 24/7. As we are removing 24/7, the excreted function, not completely, to a certain extent, it re-establishes the secretory as well as the regulatory, this one, which you can see the serum creatine goes down, C-reactive protein goes down, urea goes down, potassium goes down, serum indoxil also goes down. Ultimately, doesn't matter what the number is, doesn't matter what disease is, doesn't matter what product it is. How do you know a particular patient is getting better? Quality of life. Is the patient feeling better, eating better, more productive, more cognitive, less stress on himself or family or caregivers? I'm delighted to tell you, we have over about 5,000 customers, 85% of them tell they are all feeling much better. Okay. So we really feel good about what we have done. We plan to do some more trials. Unfortunately, I was telling him, as a dietary supplement, people don't respect it. So we are going to take the product also as a drug. Just like fish oil was taken as a drug, we are in the process of just taking a product as a drug. So it's going to cost more. Unfortunately, you know, people don't accept dietary supplement as much as they accept drugs, I should say. Even in the underdeveloped countries, they prefer drugs rather than dietary supplements, I should say. Only in USA they accept dietary supplement more. So the future of probiotics, celiac disease, autism, allergies, oral dairy, diabetes, urinary infection, all this, whatever it is, fecal transplant studies, he just addressed. I don't want to tell, this is what it is, fecal transplants. And he said, now you have the capsules. A company called as U Microbiome in Boston gives you fantastic capsules, which is totally infection-free. It is guaranteed, it's very obtained from very healthy volunteers. And most of the volunteers, they're all athletes and students from Harvard University. So he was telling you not only get a good microbiome, you probably also are getting a microbiome with probably a good genes of intelligence, I should say. So we started this. This is just to give you some sort of an idea. And, you know, as a one-man company, now we have come a long way. And we are at the point where we are actually, mostly we are an R&D company, as you know. This is Willem Jonathan Kolff. He was the inventor of dialysis machine. I had an opportunity of meeting him in 2001 or 2002. And when he started the company, we started the company with the idea of developing a genetically engineered microbe. And when I told him we are trying to take a genetically engineered microbe and the people are against taking a genetically engineered microbe, he said, Dr. Rangan, don't worry what people think. As long as you feel that you have a concept which you believe in, you think it is possible, I should say, you move forward. When I came to this country from Netherlands, working in Cleveland Clinic, when I told that you can take the blood out of the body and purify it, put it back, they all called me crazy, lunatic. They even told me, let's send it back to Netherlands, I should say. So you will get the same thing. But if you believe strongly what you're doing, go ahead and do it, I should say. The gentleman on the right is Eli Friedman. He's our chairman of our scientific advisory committee. And of course, this picture we took in our 10th anniversary. And this is probably the last but the least. When we wanted to do human clinical trials, our chairman asked me, we want to do it, well, do you have phase 1 studies? I said, phase 1? Our product is a dietary supplement. We don't have any phase 1, I should say. He said, well, if you don't have any phase 1, I cannot take you to the IRB to get the permission to run the clinical trials. Then he asked, well, I have no money to run this phase 1 studies with 50, 100 people. He said, doesn't matter. Can you run it with about half a dozen people? I said, possible. He asked me, can you take the product? I said, I can, but it is a veterinary product. He said, isn't the veterinary and human the same? I said, fine. I said, how about your wife? My dear wife is sitting there, and she also said yes. You know, not only my wife, my 5 other employees, all of us, what we did was we engaged Morgan Lewis Baucus, our law firm. We wrote an agreement that all of us are taking the product on our own will, not at the coercion. And we engaged Dr. Nicholas Keflidis, who was a professor of medicine at University of Pennsylvania, as our medical director. And for 4 months, every one of us were consuming 2 to 5 tablets. And we had our medical, we had our physical, we gave our urine, we gave our feces, as well as the blood. It was sent, and all of us were healthy. And upon that, we got all the approval for doing the IRB issues. And Dr. Friedman has got excellent foresight. He said, Dr. Rangan, you make sure to take the picture before you start, because you guys are making history. So this is the picture that we did, and we have been participating in the World Congress of Nephrology, which is held every other year. This was in 2005. This was in Singapore. We actually participated, followed by that in Rio de Janeiro. When we went to Milan, and we went from there to Vancouver. We went to Hong Kong. Recently, we went to Cape Town. So every other year, we participate. So we are slowly coming to the aspect of this. And thing is, a lot of people ask me, Dr. Rangan, how did you come up with the name Kibo? To make the long story short, I was in a meeting of the Japanese American Chamber of Commerce. Next to me was chairman of Isai Pharmaceuticals. And he asked me, Rangan, what do you do? I said, we are trying to develop a product that you can take it orally to remove the uremic toxins. He said, why are you wasting time? We already have a product in Japan. That product is, I said, yes, I know. That product is called cremazine. It's a charcoal. And the Japanese government is a drug. They give $200, and the patient pays $70, $270 a month. They have to take 9 grams per day. But when they take it, it does remove urea and creatinine and uric acid. Doesn't touch urea. But it also binds, most of the patients take other drugs. It also binds some of the drugs they are taking. So I told him, we want to make a product which is better than what you have. He said, are you sure you can make it? I said, that's our vision. That's what I'm thinking. Then he said, how did you come up with the name Kibo? I said, well, we basically named it the name Kibo. We are trying to substitute the failing kidney function by the bowel. Kidney and bowel we put together. He said, well, if you think you can make what your vision is, you will have no problem in selling your products in Japan, because in Japan, Kibo means hope. So I always say we want to give hope not only to the Japanese population of patients, we want to give hope to the entire world population of patients. And as we told, we are the first company to be nominated and won the award as a dietary supplement company in the Bus of Bio Award that was held in San Diego last year, I should say. Because Bio only accepts drugs, therapeutics, or vaccines, but we are the first company to get into this. And well, I always say what you have learned is only a fistful. What you have not is the size of the world, I should say. Thanks for everybody. I don't know whether I should tell you this. I get a goosebump. I'm from a family of 13 children. I'm the 8th one. My mom went up to 5th grade. My dad went up to 11th grade. I'm the first one who could step into college. And all this belongs to them. Thank you.
Any questions?
What is the meaning of pouchitis?
Say that again.
You said pouchitis. What is that?
Pouchitis is also a sort of a disease. I don't know much about it. Actually, it is.
It's in the largely in the large intestine, the cecum. There is little offshoots like bubbles, and they can get inflamed. It's a bit like diverticulitis. Yeah, so it's the little pouches that come out. I just want to comment, I think just a delightful presentation, and the transition between our understanding that the bacteria in your gut do more than just sort of digest fibers and things like that. This morning, I guess, we've had a chance to see that the world is about to change, and we're right on the cusp of that. And the type of thinking that you've done is critical, and innovation is always extremely difficult, but when you start to crack the nut, it's enormously rewarding. And so we do thank you.